Good morning, and I hope everybody had a good Thanksgiving, because I'm assuming most of you are watching this after Thanksgiving, even though this is being recorded before. But I look forward to seeing you on Tuesday. And remember, on Tuesday after Thanksgiving, we're going to assume that you've watched this, and we're going to discuss papers 9, 10, and 11. And then kind of get ready for the exam, which is going to be on Thursday. So thank you very much for getting ready for that. This lecture is about flavor, about meat flavor. And what we're going to talk about are the components of meat flavor and the chemical reactions involved in meat flavor. And then we'll talk about some of the live animal factors that impact meat flavor, and then talk about the effect of cooking on meat flavor. I think most of you know that Dr. Kurth and I work a lot in meat flavor because I work with the sensory lab. So flavor is a very major component of the work that I do. And flavor is a little bit more complicated than tenderness and juiciness. And some of that is because there's more than one sense involved in flavor. So when we talk about flavors, we talk about compounds that stimulate the olfactory bulb. The olfactory bulb is located at the bottom of the sinus cavity. It's involved in the sense of smell. So when you smell things, that's your olfactory bulb. There are volatile compounds that have there are receptors on the olfactory bulb. When you bring it in through the nose, they hit the receptors, and there is a stimulus that goes to the brain. It just happens that the place in the brain where the sense of smell is detected also is close to the emotional center. And that's why we associate so many smells and tastes with emotions. So for me, this is Thanksgiving season. Also where I'm from, this is pheasant hunting season. And when I smell pumpkin or pumpkin pie, I get these very nice, wonderful, warm emotions because it reminds me of time with my family, pheasant hunting, and Thanksgiving, which are all kind of tied together, and fall in Colorado. So that's an example of the sense of smell. But we also use the olfactory bulb when you are put things in your oral cavity and you start chewing. And there are a lot of volatiles that are then released as you chew, volatile aromatic compounds, as well as other compounds that may not be aromatic. When we say that, that means that those compounds may reach the olfactory bulb, but there's not a receptor there for them to stimulate, so you don't detect them as an odor. And this is the taste or the flavor that when you had to take medicine when you're little and your mom said plug your nose, that means what you're doing when you're plugging your nose is you're keeping air from moving up to the olfactory bulb. And you take it and all you do is you taste sweet, salt, sour, bitter, umami on the tongue, but you don't get those flavors. And so you can play around with that. It's Thanksgiving. You can have a piece of that nice, wonderful pumpkin pie or pecan pie, whatever is your favorite. Plug your nose and see if it tastes the same as when you unplug your nose. And you can draw air over it like that, and it will bring more of those volatile compounds up to the olfactory bulb, because that's really at the base of the throat and the sinus cavity. But we also have the sense of taste, and taste receptors are on the tongue. And there are basic tastes. Those are salt, sweet, sour, bitter, and umami. And that's where we won't talk that much in the sensory lecture, but if you take my sensory class, we'll talk about the taste buds on the tongue. And again, they have receptors for where certain compounds that are they have to be water-soluble compounds, go on the tongue, and if those receptors are stimulated, then it goes to the brain and you say, ah, sweet, ah, sour. So We have the nasal cavity and the oral cavity. We also have the sense of mouthfeel. That is the sense from the trigeminal nerve. And think about drinking very strong tea. That will give you an astringent mouth puckering. I think about the gummy worms that are the sweet and sour ones that have the powder on them. Sorry, I'm not an expert in candy, or Sour Patch Kids, that's what they are. You put them in your mouth and it's very sour on the tongue, and then after you get that coating off, it becomes sweet. That's a good example of something where you can get the basic taste. So, flavor, I'm sorry to say this, but it goes back to chemicals, goes back to organic chemistry. Don't block me out, but organic chemistry. And not my favorite class, never thought I worked that much with organic chemistry, but flavor compounds are chemical compounds that can vary in concentration and sensitivity of the receptors to the compound. And we get a lot of compounds due to chemical reactions that where we can have, flavor can change. So think about flavor as these volatile compounds kind of in a cloud that come in, that you can first smell because they're going to the olfactory bulb, and then you put in your mouth, you start chewing, you volatilize those compounds. So anything that's water soluble is going to affect the tongue and basic taste. And then that cloud kind of goes up to the olfactory bulb, and you may sense flavors. And the flavors that you detect, you may not be able to always tell one flavor from another. I can train you to do that, but or you may be very sensitive. People differ in their acuity based on the number of receptors that they have and what they actually, what senses go to their brain. So sensitivity of the receptors to the compound are very important. And you hear a lot about people changing their sensitivity to flavors as they age, as they reduce the number of receptors they have. It's kind of complicated because it isn't only what are the volatile compounds or the water-soluble compounds that hit the tongue, but then it's the number of receptors and how you perceive the sense that is stimulated by those receptors that makes up flavor. What factors influence perception? Obviously the chemical structure, what compounds are there. Not all compounds are aromatic. And Dr. Kurth has the Green Book. And the Green Book identifies compounds, and then it not only characterizes them, but those that are aromatic, it describes what kind of aromatics you can get that are associated with it. But also the degree of oxidation. We talked about lipid oxidation, and we know that we have initiation, propagation, and termination. And through that process, we're producing lots of different compounds. And I'm sure that you remember that lecture and probably may have went, oh my goodness. It's a very complicated picture because there are a lot of different compounds that will be produced. And we're going to come back to that as we talk about flavor. The initial levels that are there, that are in a component, and those have to be volatilized. You don't get all of the flavor in beef or pork or lamb or chicken until you start chewing it. You can smell some things, but it becomes more intense in flavor when you put it in your mouth and you start to chew. Because what you're doing is you're releasing a lot of volatile compounds. And there can be interactions with other compounds. We are going to talk about how basic meat flavors developed, and then we're going to talk about lipid heat denaturation during cooking. Let me give you a hint. It's just lipid oxidation, only it's applied, it occurs due to cooking. It's a natural process. Those two things interact, and some compounds will become higher in concentration and affect the flavor. So this is pretty complicated. It's a lot of fun because most of us like to eat. and flavor is a very important component. So when we talk about flavor, I want you first to segment flavor into two categories. There's the lean component, and then there's the lipid component. Now, I just said those two are going to interact, but what I'd like to do is first talk about the reactions involved in the lean component, and then talk about the lipid component, and then we're going to talk about their interaction. So what's the lean component? That's the component that's associated with the protein and water-soluble reducing sugars. So protein in the meat. And what happens is that we get water-soluble reducing sugars. They're one major reducing sugar. You're thinking, oh my gosh, what is this? Glucose. Glucose is needed for energy. It's stored in the muscle. We know we have about 1% glycogen in muscle after the onset of rigor mortis. We also have some small levels of fructose and some other reducing sugars. They're components that are involved with transcription and building of compounds. So we have, it's the sugar component in the meat, mainly glucose, but also some fructose. And then amino acids. The two main amino acids are the two sulfur-containing amino acids, threonine and cysteine, that are involved in flavor. Now, other amino acids can be involved in this, but the ones that really cause some flavor effects have the sulfur containing. So we always have free amino acids because that muscle is always doing some protein synthesis. So there are amino acids. Remember, we have the lysosome, and it breaks down subunits of proteins into amino acids, and it spits those amino acids back out into sarcoplasm, and then that's used to produce, in protein synthesis, to produce new proteins. So there's always some free amino acids in the sarcoplasm. And when you heat these two components together, they produce meat flavor compounds, volatile meat flavor compounds. Also, there is myoglobin and non-heme iron that's in the sarcoplasm. We know all about myoglobin. There's also a little bit of hemoglobin in there. We don't ever get all the hemoglobin out. The hemoglobin is going to be contained in the capillaries that are there, inside the muscle. And the non-heme iron, these are mainly related to flavors, or they assist in increasing lipid oxidation here, lipid components, and but also muscles that have more myoglobin are higher in some flavors, tend to be more serumy, bloody flavors, metallic flavors. We also tend to get the development of liver-like flavors in some situations in those muscles. And those are associated with this non-heme iron. What's non-heme iron? That's iron that's free, that's not part of that heme and myoglobin. We always have a little bit of free iron because we need to build and replace myoglobin. It's a very key part of the homeostasis in that muscle fiber. So we know this is the main driver of flavor. This is kind of a minor component, but it can have some effects. So the lean component, but then let's look at the lipid component. We know the lipid component is either from the triacylglycerides and there's going to be some free fatty acids that are floating around in the sarcoplasm. They're going to be used and broken down in the mitochondria for energy. And we're also going to have triacylglycerides in the adipose tissue. Remember, we're going to be bringing some of those triacylglycerides into the muscle for energy. So there's going to be some presence. And then we also have the phospholipid component in any of those membranes, in the sarcolemma, in the membrane of the sarcoplasmic reticulum, in any membrane that's there. So the lipid component is associated with the species-specific flavors. and it is related to the amount and the total amount of fat and the fatty acid composition. If you go to the USDA Nutrient Data Bank online, and you go to Handbook 8, and Handbook 8-13 is for beef, 8-10 is for lamb, and I think it's 8-6 is for pork. Sorry. Can't remember, but they're in there. And you look at the fatty acid composition of the longissimus dorsi, which is, of course, the main back muscle that makes up the loin and the ribeye. And you look at the fatty acid composition, you see that lamb, pork, and beef differ in fatty acid composition. And the differences in those fatty acids, while it may not look like it's that much, affect the flavor. Lamb flavor is different than beef flavor, is different than pork, because from a species standpoint, not the lean part of it, but just that you say, oh, this tastes like lamb. That's due to differences in fatty acid composition. But we also know that as we cook meat, lipid oxidation occurs. And when we're cooking, we call it lipid heat denaturation. But it's the same reaction. It's the use of heat to cause the lipids to denature and go through the oxidation process. And so depending on the initial level of lipid oxidation, and we're actually studying some of that in my lab. Anderson Cabral is looking at that as part of his dissertation. We're actually creating differences in the LD muscle and level of initial lipid oxidation. So we are going to have T-bars of basically 0.1, T-bars of 0.5, and T-bars of 1. And in T-bar levels, a T-bar level of 1 in raw meat is pretty oxidized, okay? So we have no oxidation, medium oxidation, and lots of oxidation. And then we're going to cook that meat two different ways. One, so that we don't get that much of the lean component to cook. We're not going to get the Maillard reaction. We're just going to get mainly lipid heat denaturation. And we're going to look at the differences in flavor that this actually causes prior to cooking. So the amount and stage of development of lipid oxidation, you can imagine if you're from a raw meat in the middle of propagation, you have some termination products, and then you cook it and you speed up that whole reaction, obviously you're going to get more of the secondary products from termination, more so in those products that have higher initial lipid oxidation. I'll have more information on that next year when I give this lecture, because I'll have some of Anderson's data to be more specific. There can also be diet-specific compounds that are in the fat. What we used to think is that when animals were fed grass, and if they ate grass that was high in keratin, that keratin gets deposited in the animal. We actually found out that we used to think it was deposited directly in with the fat lobe of the adipose tissue, but those compounds, keratin is a water-soluble compound, and it does get deposited in the adipose tissue, but it's in the water-soluble component, which is the cytoplasm in the adipose cell. So remember how the adipose cell looks like this, and it has the nuclei on the outside and the cytoplasm squashed around the lipid droplet. Keratin would get deposited in there. Keratin is the most common compound that's deposited there associated with grass feeding, and it gives us yellow fat. We know keratin is what gives carrots their orange color. It also, we don't get as much keratin deposited in animal fat as in carrots, but we get some. And when you see yellow fat in poultry and And we don't really see it in pork because we don't grass feed that much pork, but in cows, especially in carcasses from older animals, it's due to that compound being deposited in the adipose tissue. There are other compounds that can be deposited there too that are diet specific. Most of them are associated with forages, with grasses, because with animals that are fed grain-based diets of corn, wheat, barley, sorghum, those compounds aren't present. So most of the compounds that we're going to talk about here are associated with grasses. And I gave you the Melton paper. We're going to talk about that in a little bit more detail here, where they looked at different grasses and forages and their effect on flavor and meat. One of the kind of interesting things is that there are places in the United States, mainly in North and South Dakota and Minnesota, where the soil is very high in selenium. There are some plants that concentrate selenium. Animals don't like to eat those plants, and the reason is selenium doesn't taste very good. And if you want to see if that's really true, go to the vitamin section of the retail store, look at selenium, and you can you probably can't open the lid, but if you want to really do this, you could buy that. Maybe I should bring some in so you could smell it. But selenium doesn't smell very good. My husband made me take selenium supplements for a while because they're associated with improved brain function, and they've been shown that people who have potential for onset of Alzheimer's, it reduces or delays the onset of Alzheimer's. The ironic thing is that that's in my husband's family, not mine, and he said he was just protecting me because my memory was already better than his and he needed me to keep it. So, but I stopped taking those because I couldn't stand the taste and the aftertaste of them. Well, animals don't like to eat that grass because of how it tastes. But in usually August and September in that part of the country, the grasses that the animals like to eat are gone, and they have no choice. So they eat the selenium-based animals or the grasses, and those compounds they actually deposit in the water-soluble component of the adipose tissue. And we've smelled claybird up really bad by cooking that meat. Companies like McDonald's, Burger King, Wendy's will not buy lean trimmings from that part of the country during late summer and early fall, because that's when they get this real specific off flavor associated with it. So that's one of the best examples of diet-specific compounds, carotene and also those selenium-based compounds. Also, there are hormonal or sex effects on meat flavor that are associated with testosterone. And we'll talk about that especially in pork. We also can get flavor from microbial growth. We know that as microorganisms grow, as foliage organisms grow, we're going to have some off flavors. If you have aerobic microorganisms, those foliage organisms are going to produce sulfur-based compounds. That's why meat that spoils when it's left out in the air, think about Vietnam. We had that discussion on Tuesday, losing track of days. Those are aerobic foliage organisms, mainly Pseudomonas. Dr. Taylor probably talked about those, hopefully. And those microorganisms use the proteins from the meat, and they produce sulfur-based compounds, and they smell like rotten eggs. Very pungent. Off flavors from anaerobic organisms are usually lactic acid based, because those are lactobacillus that produce lactic acid, and that's why we get sour milk flavors associated with spoilage in vacuum packaged meat. There can be some other degradative processes that are proteolytic that might give ammonia or sulfur-based type of aromatics. But then cooking also affects flavor. And we have two major reactions that occur in cooking. One is the Maillard reaction, which is associated with this lean component, and then the denaturation due to heat volatilization of compounds in the lipid component. So I hope that kind of gives you a good overview. What I'd like to do is get into each of these issues with a little bit more detail. I gave you two papers in your folder. I gave you I just went blank. I gave you the Melton paper, and then I didn't give you the Wasserman paper. I gave you the other paper, which I'm sorry, I'm still kind of fuzzy from the Vietnam trip, but which is a review. And in that other paper, it talks about the Hornstein and Wasserman paper, where they were the first people to really get that basic meat flavor and to understand it. So what they did is, first of all, they took fluids from raw beef lean, and when you smell raw beef, it smells ceramy. If you eat raw beef, which I wouldn't encourage you to do, unless you know that it's microbially safe, you eat it and it's really kind of ceramy. It doesn't have any of those meat brothy type flavors, any of the salt flavors. It's a little bit sweet and ceramy. And what they found is that if you took the fluids from raw beef, you had to cook them to develop typical meat aroma. And if you took the components of meat and you separated them, so you separate the lean into the sugar components, the amino acids, and other lower molecular weight polypeptides, do this by centrifugation, that when you heated the components separately, you didn't get lean meat, that you had to heat them together. And that means that there had to be interactions of components. And we know that the fluids from lean meat contain sugars, mainly glucose, some glycogen as a component of glucose, and some fructose, also salts. We have sodium, we have potassium, those type of things. Amino acids, and then there are some low molecular weight polypeptides that are present as building blocks. And so what they found out is that you have to heat those components in combination to get typical meat flavor. And what they found was that meat extracts with high amounts of amino acid-like compounds and carbohydrates ended up with more intense meat-like flavor. A lot of this work was done early on. It's done in the 60s because they wanted to develop beef flavor compounds like beef broth, and trying to understand how they could do that without making meat. And so we do have those type of things that are available. But you have to have the amino acids and the carbohydrate to get the meat flavor. So that's where the lean component comes in. And so during cooking, with the use of heat, amino acids and carbohydrates interact to give us meat flavor. But then when they looked at meat from different species, and they looked at animals of different maturity and fatness, they found out that they differed. So that it wasn't just the lean component contributing to flavor. And actually, if you take meat from goat, which differs in flavor than meat from beef and pork, they found that from a sensory standpoint, consumers found it less desirable, and it was less tender, right? Less satisfactory overall than beef, pork, and lamb. And to understand why, they found out that it was really through the genetic control of lipid composition and metabolisms, which is basically fatty acid composition, that contributed to that differences in flavor. So we still have the lean flavor development, like in meat like goats or in any of these, But it is the lipid component that differs. Even the phospholipid component differs somewhat on what fatty acids are attached to that phospholipid, and that it's the heat denaturation of the lipid component that gives the species-specific flavor. So the paper I put, the Mortram paper, I think is the one I gave you, and, sorry, yeah. So the next component, so we know the lean component, we know where the species-specific component comes from in flavors, but we also found out that meat from bulls tastes different than meat from heifers. Now, some of that is the amount of fat, just total amount of fat. So you get more lean and less fat, and you get less lipid heat denaturation because there's just less fat. But with increased muscle growth, we also found that meat-like flavors sometimes had these off flavors. And those were associated with byproducts of testosterone. And we see that actually in boars, in pork, and lamb and beef. The main difference in sex effects is due to that decrease in total fat and increase in meat-like flavor. So the boar odor, and what's interesting about boar odor is that boar odor can actually be found in sows as well, because there is some testosterone produced in sows, but there's a lot of testosterone produced, obviously, in boars. And that boar odor is associated with 5-alpha androstenedione, basically it's a metabolite of testosterone, and skatol. And this is a very urine-like odor. There are some people that can't detect it. It's very interesting that some people have no ability to even detect it. But if you can detect it, I can detect it very easily. You can smell it on the carcass. If you heat the fat from those animals just a little bit, it can smell up the whole kill floor, let me tell you. So it's a urine-like sweaty aromatic odor that develops when boar fat is heated. And it's either a breakdown product of testosterone or a secondary product produced in the animal system related to testosterone metabolism. We know that skatol levels are a product of testosterone. Not every boar has boar odor, but there is a much higher incidence in boars. And some of the work that they've done with doing immunocastration of boars as young animals so that you don't have to physically castrate helps to reduce the boar odor, but we actually see immunocastrated animals versus castrated animals. We'll still see a little bit higher level of boar odor in the immunocastrated animals. We can also detect some of this from gilts in castrated males, but as long as we're harvesting those animals, and in pork in the US, we harvest right about the onset of puberty. So we don't really see a high level of it in castrated males or gilts in the pork industry. If we feed those animals beyond that, we'll start seeing some incidence of it. And where it really shows up is in cooking. And if you have bacon that's produced from older animals and you have boar odor, you can detect it. As you cook that bacon, because we tend to cook bacon at higher degrees of doneness, and it has a higher level of fat, it's very aromatic. And my husband can't detect it, but I can, so it's kind of interesting. When we look at castration for rams versus wethers and sheep, it has little or no effect on lamb flavor, depending on the age and time of slaughter. Again, we tend to slaughter our lambs in the United States just a little bit after onset of puberty, and we castrate most of our rams. We don't really kill rams in the US, but in other countries they do. And as increase of time past puberty gets longer, then rams tend to have more intense flavor, and it's more of the intense lamb flavor. So we were doing some work with lamb flavor for some collaborators at Montana State and also at San Angelo. And so we've been doing a lot of flavor work with lamb. And one of the things that we try to do is to get lamb meat that has off flavors, to give that to the panelists so that they understand what that's going to be like. And if we could find some meat from an old ram, it'd be wonderful. But they tend to not be slaughtered in the meat production system. And when we have wethers and ewes of the same age, we really don't see any differences in flavor. They will have the lamb flavor, but we don't really get the development of the muttony flavor, which is a kind of a musty, urine-y, dirt type of aromatic. But we will see the onset of that in rams a little bit earlier in age than in wethers and in ewes. So there is some sex effect, but we don't tend to have those animals in our production system. For bulls and steers, as long as the bulls have not shown a lot of secondary sex characteristics, meaning that they aren't that far, they either at the onset of puberty or just past onset of puberty, we see very little effects. But if we slaughter our steers and heifers past the onset of puberty in the United States, because they're going to have onset of puberty at about 12 to 13 months of age, and we slaughter some animals that age, but most animals are a little bit older than that. So if you have bulls that are showing secondary sex characteristics, we tend to see more off flavors associated with those bulls. Some of the reason is they have higher myoglobin content. They have higher myoglobin content because they have more muscle development and less lean, and also they're getting, ready to fight and protect their herd. The flavors that we see that are associated with higher myoglobin are livery flavors, grainy, cowy, metallic, serum-y type flavors. They also have less fat. So if we have less fat, we see more of those meaty flavors and less fat flavors. And what's interesting, in the United States, we're used to eating beef with some beef identity, beef brothy, beef lean flavor, and a little bit of fat. And when you change that ratio, people don't always know, well, this has more lean flavor and this has less fat. They just know that together it tastes different. And we know that testosterone is involved in that because it's involved in producing less fat and a higher muscle development. Also, bulls have been shown to be more susceptible to long-term stress pre-slaughter. It's more DFD. And when we have DFD meat, which is higher in pH, it tastes different. And we're going to get more of the musty, moldy, or more intense beefy flavors. We'll get more cowy, grainy, serum, bloody flavors. So it tastes just more intense in flavor. And for those of you who are saying, I wish I knew what these flavors you're talking about, you may think, they may induce some thoughts in your mind. Take my sensory class and we'll talk more about that. My sensory class is in the spring. I know some of you are taking it, and I'm excited about having you in class. We do know that meat from older animals is more intense in flavor than meat from younger animals. We talked a lot about that as far as differences between young and old animals as it's associated with tenderness and the palatability puzzle. But from a flavor standpoint, we know meat from older animals is higher in cowy, grainy, serum, bloody flavors, and more metallic. And they also tend to have less fat flavor because they tend to not be fed to the same endpoint, right? Because from a production standpoint, feeding high concentrate diets and getting more marbling in those animals is not economically conducive. We'll also get more, the serum, bloody, and metallic descriptors have been also associated with higher levels of myoglobin. So not only as we get meat from older animals, they have more myoglobin, the meat is also going to have lower in fat, and we see this shift and change in these attributes, and it tastes different, and consumers actually can tell that. In sheep, we get more mutton flavor associated with meat from mature sheep, and if you don't know what that is, we need to go over to Pierce Pavilion and look at some of the old, have you smell some of the old fleeces from older animals. I did that with my panelists, and you can pick up the mutton flavor just like that. Some of you may know what that smell is, some of you may not. We noticed that the diet that older animals are fed are usually roughage-based. We talk about roughage-based diets having a higher likelihood of having some of those compounds that are deposited that may affect flavor. We have more problems with yellow fat with meat from older animals because they're forage fed. And there's been a lot of work that's been done about forage-based diets and how some of those flavor attributes get deposited. So we know that happens. In lambs, meat from animals fed roughage diets had lower flavor scores than lamb-fed high energy density diets. And again, in lamb we see a lot of that's due to fat differences, right? And we know that not only the total amount of fat, but the fatty acid composition affects that flavor development during cooking. Because we have more lambs that are roughage fed, in California, most of the lambs that they kill come off of forages because they have very good forages through the spring of the year, and those lambs grow and come to slaughter weight very rapidly and may not have any grain feeding, and therefore they have more susceptible to those compounds being deposited in the adipose tissue. When we feed high concentrate diets, it takes about 70 days for fat to turn over. So when feeding high concentrate diets, after 70 days, we'll see the disappearance of those off flavors that are associated with the roughage, because grains just don't have those type of flavor compounds to be deposited. I gave you the Melton paper. Sharon Melton was at the University of Tennessee. She's since retired. She did a lot of work on roughages and its effect on flavor. And I'd like you to look at that paper. I've kind of summarized a lot of the big things from that paper here in the next couple of slides. But what Melton found was that high concentrate finishing diets resulted in the most desirable flavor. Now I want you to know that she summarizes a lot of the work up through 1983. Really until the last 5 years, we haven't had a lot of research since the early 80s on flavor. Now flavor research has really increased in funding, and we're really looking at trying to understand the chemistry of flavor to a greater degree. And so we're using a little bit different tools than what Melton had available to her when she was doing this research. So her sensory work would look at just overall flavor intensity and positive flavor versus like lean flavor versus off flavor. And what she found was that corn, barley, wheat, sorghum based diets are what she's talking about when she talks about high concentrate diets, that those produce meat with the best flavor. And that you can partially replace or totally replace those corn, wheat, barley, sorghum-based diets with corn silage and have almost no effect on flavor. Corn silage and alfalfa based diets had little effect on flavor. Alfalfa hay any combination with alfalfa and Timothy hay. But when you use other forages, they saw a change in flavor. And she talks about that most of that first change that they detected was due to lower fat levels, lower just total levels of fat, and they were not looking at specific flavor attributes because the beef lexicon hadn't been developed. They didn't use flavor descriptive attribute, but she talks about how when you're feeding forages and replacing it with high concentrates, the first thing you do is, because you reduce total fat, you see less desirable flavor. But there are some forages that also change flavor that are associated with those flavor compounds. And those that are associated with changing flavor, she talks about brome grass and blue stem, bluegrass and clover, fescue, orchard grass, clover, fescue alone, Flint Hills grass, native range grass, forage sorghum, I don't know what that is, and rye. And then ryegrass, millet, coastal Bermuda grass, and Sudan can impart off flavors, but these they found differences in flavor. So you can see that I'm not going to ask you specific forages that affect flavor, but that forage level can really affect flavor. When we talk about grass-fed beef, and you can go and get Australian grass-fed beef at HEB. Nolan Ryan did have a grass-fed beef program. They've since disbanded that program for economic reasons. There are other places that have grass-fed beef. A lot of people are talking about grass-fed beef. One of the biggest challenges with grass-fed beef is that forages don't have the same maturity and quality all year round. So this is the rule of thumb when you're talking about forages. If forages are young and growing and animals are eating them and average daily gain is good, usually off flavors are not associated with that. But once forages mature and the animals are having to scavenge more and they're not growing as fast, we tend to see more of the off flavors deposited in the adipose tissue and off flavors come out. So I do a lot of programs for the extension group, and either Beef 101 or Beef 706. One thing we do with the flavor component of that program is we give them grass-fed beef. Sometimes the grass-fed beef is wonderful and has no flavor issues. Sometimes that's not true, and it can be very intense to not very intense. They may pick up on it, some of them may pick up on it, might be slight. Sometimes it's like bam. It's there and it's very intense, but it might not be the next time. So the biggest challenge we have as meat scientists with grass-fed beef, lamb, or pork, sorry, I'll turn that up. Is that we don't always know when we're going to have good flavor and when we're going to have off flavor. It's the variation in flavor. So that's what Melton's talking about. So what are some of the strategies to change grass-associated flavors? One of the things that we've found, we've done some work with this, is supplementation of grains at a very low level while they're on pasture, usually about 2 pounds per head per day. We've found we'll kind of reduce the development of off flavors because the animals are having, they're growing well, they're eating forages, but they have some grain supplementation that reduces forage intake, so we tend to not get that concentration of off flavors. And then if young cattle are removed from pasture and fed a grain-based diet for 90 to 100 days, it's really 70 days, which is the minimum. So normally we say 90 to 100 days. Prior to slaughter, we can reduce those grass-fed off flavors in the meat. And pork's a little bit different because they're non-ruminants and they are what they eat. So whatever you give the pork, they're probably going to taste like it. And there are some very famous programs that feeding acorns in Spain and Italy result in pork that has a nutty flavor. Imagine that. Feeding pork apples will give pork more of an apple-like flavor. They are what they eat. We know in the U.S., we basically feed pork corn and soybean-based diets. So we have very little effective diet on flavor in young pork. We have these, we do have some programs, some niche market programs that have developed. Nyman pork is one of the free-range programs, natural programs, that's sustained itself over a number of years. They're, one of their quality control people came to Texas A&M, and we did some cross-training for sensory, because one of the biggest problems they have is they have different ranches that or farms that are producing their pork, but their forage may be different, and the variation in flavor is one of their biggest challenges. So depending on what we feed pork, it's going to taste like it. And we know that in ruminants, the microflora bihydrogenate the unsaturated fatty acids in the ruminant, and that's why we get more consistent fatty acid composition, and we don't really have a change. But in pork, they are what they eat, so we can actually change the kind of oil that they eat and change their fatty acid composition. That will affect their flavor, as well as any specific flavor compounds that might be from their diet, like the acorns or the apples. It's kind of fun to play with this. So, let's talk about cooking on meat flavor. When we cook meat, we cook meat to different degrees of doneness. And we talked about, when we talked about color, how that change in color in meat during cooking is associated with rare, medium, medium well done meat. It goes from red to gray. That's denaturation of the myoglobin, heat denaturation of myoglobin. And as we look at that meat and it changes in degree of doneness, meat that's cooked to lower degree of doneness is going to have more serum bloody flavor, and meat that's cooked to high degrees of doneness is going to have more roasted, more cooked beef brothy, more beef identity, and less of the serum bloody. And consumers pick that flavor differences up. They may not know what those specific attributes are, but they know the flavor differences. So we do know that degree of doneness has a big effect on flavor, and it's associated with myoglobin denaturation. During cooking, we also have the Maillard reaction products, and that's called non-enzymatic browning. And Maillard reaction occurs once we hit a higher temperature, and it's associated also with dehydration of the surface, where we get the brown burnt flavor. Brown first, and then it goes to burnt. And we can also get heat denaturation of other proteins. And then we also get acceleration of lipid oxidation. So up there are 5 points up here that all affect flavor. The myoglobin denaturation, Maillard reaction, dehydration of the surface, heat denaturation of other proteins, and accelerated lipid oxidation. We normally talk about the Maillard reaction and accelerated lipid oxidation as the two main reaction groups that occur, and then the degree of doneness and the basic myoglobin denaturation is somewhat related to how much of each of these reactions can occur. And so, how do we measure meat flavor? We measure meat flavor in basically 2 different ways. Either with sensory, either trained or consumer panels, and the next lecture I'm going to give you the Tuesday, last day of class after exam C, will be on sensory. And in sensory, if we're using training panel, we may measure the odor, the smell of the meat prior to consuming it, flavor aromatics as you chew it, what flavor, volatile flavor aromatics go up to the olfactory bulb, basic taste on the tongue, mouthfeels like that astringent mouthfeel I discussed earlier, or aftertaste that come after swallowing. We don't ask consumers these questions. We ask them what they like and dislike. We'll talk a lot about the differences in using those two different tools. So sensory is a very important part because flavor is associated with the major sensory attributes. We also can use headspace analysis. This is where we put meat in a glass jar or in some type of container, glass usually, something that's not going to impart flavors of its own. And we gather the volatile compounds that come off the meat in what we call the headspace, which is just the area above the meat. We use a SPEME, which attracts the all the volatile compounds, and then we can put that into a GC, which is going to separate those compounds. And if they have aroma associated with them, we can smell them on a sniff port and identify those that are aromatic. There are going to be some volatile compounds, as you know, that are not aromatic. And we typically use the GC mass spec system with an olfactory port to identify those. And if you are interested in this, Dr. Kurth is almost running those daily now with the number of work that we do. What we do here at Texas A&M is Dr. Kurth and I really try to combine both of these tools to try to understand what's going on. What this does is helps us to identify the specific aromatic compounds. So on that chart for lipid oxidation, where we add all those different compounds, we actually measure and have a quantitative evaluation for different compounds. And there can be over 400 volatile compounds, volatile aromatic compounds found in meat. In some of the studies, we may get that down to 180, 140 that are really important. And some of those are aldehydes, some are ketones. We'll get pyrazines that are a result of the Maillard reaction. And so we can then quantitate differences. I haven't gone into a lot of detail on the flavor reactions because we expect that you will get those in additional classes. But what I've tried to do today is help you to understand the components of meat flavor and what chemical reactions are involved in meat flavor, and then understand some of the animal factors that impact flavor development, and then understand how cooking affects meat. So thank you very much, and I hope you all had a great break, and I look forward to seeing you on Tuesday.